Tests for proteins. The contents for this video are introduction, results, discussions, and lastly, conclusion. The content. Alright, let's get started. There are various tests for proteins, such as Zendaprotic, Million test, Sakaguchi test, Burette test, and Ninhydrin test. The first test that we will discuss about is Burette test. It is used to indicate the presence of amino acid residues of peptides containing 2 or more amino acid residues. Burette test is based on the ability of copper-2 ions to form a violet-colored chelate complex with peptide bonds in alkaline conditions. However, Burette test is limited to the peptides with the chain length of at least 3 amino acids, and the length are necessary for a significant and measurable color shift. For this experiment, we achieved bovine serum albumin and casein for positive result. Bovine serum albumin is derived from cows, and the full-length BSA precursor protein is 607 amino acids in length. And for casein, is commonly found in mammalian milk, which contain a fairly high number of proline residues. Glycine is the amino acid which has hydrogen as a side chain, which acts as the smallest amino acid. Glycine does not contain the chain length with at least 3 amino acids. Therefore, it gives rise to negative result. For urea, there are only 1 peptide bond present. For proline and asparagine, this is due to insufficient amino acid chain length to form at least 2 peptide bonds. Therefore, violet complex did not form. Next, we will look into Ninhydrin test, which is used to detect ammonia or primary and secondary amines. When Ninhydrin, the oxidizing agent, reacts with these free amines, a Ruhrmann purpur is yielded. All primary amines and ammonia react similarly, but without the liberations of carbon dioxide. The amino acids proline also react with Ninhydrin, but they give a yellow color complex instead of a purple one. Besides amino acid, other complex structures such as peptide, peptones, and protein also react positively when subjected to the Ninhydrin reaction. Proline produce yellow color complex when it reacts with Ninhydrin. It's somehow special that it forms a ring structure due to R group bonding with the amino group, so the nitrogen is not free to react with the Ninhydrin. That's why it gives yellow color complex instead of a purple one. For our experiment, glycines and asparagines give rise to positive result. The Ninhydrin oxidizing agent will oxidize the free amino acid and will be producing ammonia, carbon dioxide, aldehyde, and a reduced form of Ninhydrin. The reduction product obtained from Ninhydrin then reacts with the ammonia and excess Ninhydrin to yield a Ruhrmann purpur. For bovine serum albumin, it gives rise to negative result. There are no reactions of BSA with Ninhydrin. This might be due to the absence of the free amino group to react with the Ninhydrin. Xanthoprotic test. Xanthoprotic test is a technique used to identify the presence of amino acids with an aromatic ring group. Examples of the amino acids with aromatic ring group are phenylalanine, thyroxine, and tryptophan. Concentrated nitric acid is used in this test, and with the presence of concentrated nitric acid, the aromatic phenol ring will actually be nitrated to give rise to yellow color. At alkaline pH, the color will change to orange due to the ionization of the phenolic group. Glycine, lysine, and phenylalanine demonstrated negative results for this test, while thyroxine, tryptophan, BSA, and casein demonstrated positive results. Phenylalanine gave rise to negative results. The reason was probably due to the benzene ring of phenylalanine was not activated. Thus, the ring was not readily undergo nitration. Since the ring was not nitrated, no change in color was observed for phenylalanine. Moving on, we will look into Million test, which is specific for thyroxine. In Million test, the phenyl group of thyroxine is first nitrated by nitric acid in the test solution. Then, the nitrated tyrosine complex mercury 1 and mercury 2 ions in a solution to form a red precipitate or the red solution to yield positive results. Proteins that contain thyroxine will therefore yield a positive result. For our experiment, glycine, BSA, and casein give rise to negative result because thyroxine is not present. Last but not least, Sakaguchi test. Under alkaline conditions, alpha-naphthol 1-hydroxyl naphthalene reacts with a mono-substituted guanidine compound such as arginine, which upon treatment with hypobromide or hypochlorite produces a characteristic red color. The guanidine group is a carbon group with a single bond to 2 nitrogen atoms and a double bond to 1 nitrogen atom. The Sakaguchi test contains sodium hypobromide, which is an oxidizing agent. The reactions of the oxidation with the guanidine group is what gives the results a reddish color. An excess of alpha-naphthol produces a yellow color. However, the quantity of alpha-naphthol that are required to give complete color development for arginine gives only the faintest trace of yellow. This is observed from testing lysine and urea. The greater the excess of hypobromide used in developing the color, the faster the fading occurs. The red color had faded to light pink color. In conclusion, the test carried out was specific towards the amino acids of interest. This confirmed that the presence of unique side group in the structures of amino acids based on to the unique characteristics of their particular amino acid, and this in turn contributed greatly for discovering methods to sequence a particular proteins of interest. Thank you for watching.